Okay, so yes, we started in this course with bacteria, and then we moved on to protists. These are all unicellular organisms. And you might be wondering, well, the cell is the smallest unit of life. How did it come about? The cell is already pretty complicated. Would you agree? Right? The cell is fairly complex, got a plasma membrane, all those organelles. It can metabolize, it can perform all the functions of any multicellular life form. Really, the smallest unit of life. So we're going to look at that. How did these small but not insignificant cells arise in the first place? And then we'll look at some progressions in life forms, some extinctions, and also some geology. Biodiversity. What is it? Biodiversity. It's simply the diversity of life. The diversity of life. Bio means life. Diversity on Earth. Why is it important? It's important because it maintains the geology of the Earth. It maintains the atmosphere of the Earth. It maintains all life forms when they work together in ecosystems. So they're quite related and dependent on each other. So we're going to talk about that. Phylogeny, how do you make the tree of life? And then we'll continue looking at that later. We'll especially look at the origins of today's diversity and the macroevolution from protobiont to multicellular eukaryotes. Is that a small leap? No, it's a huge leap. But fortunately, we've had 3.8 billion years to make that leap. And life is a continuum, extending from the very earliest organisms, those small bacteria. Well, the first bacteria were archaea, to the variety of species that exist today. So we can put all that in a tree, but this is a crazy kind of early tree of life where Menschen is people in German. They look like in this tree, they look like, oh, they're the top of the epitome, like the epitome of evolution. And everything else came before. But as it turns out, humans are not the epitome of evolution. Everything that's alive today is the epitome of its own evolution. So this is an enormously complex tree. You don't need to memorize this, not to worry. But these are branches. These are all branches of different groups. These are all branches of the plantae. These are branches of the fungi. These are echinoderms. And hemichordata, here are the chordata. So all of these branches are alive today. They contribute to biodiversity today. So we're really not very special. We're just part of what's been happening for the last 3.8 billion years, essentially. And somewhere on that tree is Nemo. Let's see. Oh, there he is. Sorry. I couldn't help but putting poor little Nemo in there. And we found him on the phylogenetic tree. So biodiversity, of course, includes all life forms, even like domestic forms like cats and dogs, forms that are in trouble like the panda, forms, some have gone extinct, mammals, cyanobacteria, algae, plants, protists, multicellular algae. We're all part of an enormous amount of diversity on Earth. And we break down into three major groups, as we know: eukarya, or eukaryota. That's us. We're eukaryotes because of the nature of our cells. It includes fungi, animals, plants, and some of the protists, alveolates, rhodophytes, some of the flagellates. Another domain, so these are all domains. The largest grouping of organisms on Earth, this is how they're grouped based on their cellular composition. Archaea, there's still loads of archaea in places like hot springs, of course. They include such bacteria as halophiles and thermophiles. So those are our domains. And we did look at this chap, Carolus Linnaeus, as I was trying to remember his name before he used binomial nomenclature to name species. So of all the diversity, somebody had to name them. And it was his name before he started that was Carl von Linné, interestingly. He changed it to this Latin term, Carolus Linnaeus, because, hey, binomial nomenclature was his baby. So I guess he could change his name to whatever he wanted. Yeah, and he turns out to be on a kronar, which I think is Dutch. Our kronars is Dutch, aren't they? Yeah, so there he is, honored on the 100 kronar bill. And the names that he chose are handles that convey information, like Ulmus rubra, for example. It's a kind of alder, and it's red, rubra. So there is a lot of information in Latin names, lucky for us. And he had to name all the biodiversity, the whole tree of life. That is what biodiversity is. It's a magnificent genealogy or relatedness connecting all organisms alive today. That's the tree of life. Two main lessons for us, not just students, but for me too. We are all related. Yeah, not just in the John Muir sense of all being part of the same worldwide ecosystem, but Chameleons, but literally genealogically related. And really that should inform our moral treatment of other living things. So we seem to be a little bit lackadaisical and about conservation, but at the end of the course, we'll talk a little bit more about conservation and why we should conserve organisms that may be going extinct. I think we have a moral and ethical obligation to do that, but there are other reasons, including the health of the planet. And also, we're not all that special. We're just one tiny twig on this gigantic tree. Here we are. This is us here. And there's the gigantic tree that took root on one little planet way back in time. So what is the value? Well, there's an ethical value, isn't there? Each lineage is a thread in an heirloom fabric that we have the responsibility to pass on to future generations. It has intellectual value. I don't know about you, but because you're in this course, I assume you're like this too. You have a need to understand the world. That's what education is all about, understanding the world, how it came to be, and where we fit in. We have definitely an ecological responsibility, and there's value in diversity for the proper function of ecosystems. And of course, it's the raw material for natural selection. If species start disappearing, there's less and less variation to select from for the future. The gene pools become smaller and smaller. And economic, sure, there's economic value. There's a potential source of all kinds of products of direct economic benefit, medicines, food, aesthetics, shelter, all of that. You know, when I was in Costa Rica, that was a long time ago now, in 93, I think. I don't know if I told you this, but there was some researchers there that were studying only 3 trees, and they found 200 species of ants that had never been described before, only in those 3 trees. So there is a lot of undiscovered biodiversity that could contribute quite a bit to economic as well as ecological benefits. So here's the eukaryotes, the metazoa, that's the animalia, that's us. The fungi, which include mushrooms, molds, and yeast. The green plants, which derive from green algae and land plants. The rhodophyta, that's a red algae. Alveolates, dinoflagellates, ciliates. This is a bit of a abbreviated kind of eukaryote tree. The straminophiles, which includes our brown algae. Diatoms, oomycetes, and chrysophytes. And we've looked at all of those. We looked at all of those in our labs already. Not the fungi and the plants, but the algae and the metazoa. You know, every term up to now, I've never been this far along at this time of the course. It always takes a bit longer to do everything. We rarely get to fungi and plants, but hey, we might be able to do that in this course, which would be cool. So how do we discover phylogenetic history? You can't see it. You can't see phylogeny. We're guessing who our ancestors are, basically. So how can you make inferences about it? Say you had a huge oak tree and it's buried in a sand dune, and only the tips of the twigs are showing. Just the tips of the twigs. The rest of the tree is underneath the sand. What would you do? Well, we try to find in fossils the concept of characters that are historical markers. Certain characters that changed its condition along a lineage. Like breathing apparatus, for example, lungs, or circulatory systems, like a closed or an open circulatory system. We reconstruct our evolutionary past, our phylogenetic past, based on characteristics. We look at extant species and go, yeah, cats have retractable claws and dogs don't. So that's a distinguishing feature among them. But there are also features that appear in a more complex fashion later in the phylogenetic tree. So we try to piece it together. And, you know, it ain't easy, really. Basically, all phylogenetic trees are hypotheses. And we're going to look into phylogeny in the next chapter too. So we need to find markers, a changing state, something like lungs or hair, the advent of hair or feathers. So some marker that must have changed at some time in the past. And that way we can follow or try to build our phylogenetic tree. So this is a phylogenetic tree. And these are the species, not showing any species on here. And this is where there's a shared history among these two species. They share a history there. And there's some kind of marker of a changing state. So in phylogenetics, we're going to look at homology. It's a feature that's shared by two lineages because of descent from an ancestor that had the feature. And up to now, we've looked at things like, what did we look at? We looked at the radius, for example, is found in cats and humans and whales. A transformation is a change, a heritable change in homology from a prior to a posterior state. Divergence is the splitting of one lineage into two lineages. Reticulation is the blending of two lineages into one. That's when you have hybrids. Monophyly is all and only descendants of a common ancestor. So when we get to phylogenetics, phylogenetic terminology, we will address these things more closely. So when we're watching the film today, keep in mind that we're looking at characters of extant species and characters of extinct species. Where did life evolve? So it's odd. Look, we're going to look at the origins. That's the important thing today. What's odd is that geology changes life and life changes the planet. So, and I was just watching something today about how, you know, what was I looking at? I can't remember, but it had something to do with water. So water and glaciers and the breaking up of certain minerals and the releasing of certain gases from parts of the earth. So the Earth's atmosphere and geology changes with the advent, for example, of photosynthetic organisms. Oh, the atmosphere all of a sudden became a lot of oxygen that hadn't been there before. But very early on, so the Earth formed 4.6 billion years ago. Early atmosphere had lots of water vapor and lots of chemicals released by volcanoes. Some organic compounds may have come from space. There's a lot of stuff landing on the Earth from space all the time. Small meteors, and I think they call it space dust. Yeah, there have been some carbon compounds found in some of the meteorites that have landed on Earth. But in the lab, it has been shown that the early Earth atmosphere, as far as anybody knows what it was like back then, and you can tell because what we look at is isotopes in ancient rock. So we can extract from that the composition of molecules in the Earth, even 4 billion years ago. So it turns out that organic molecules can be produced from inorganic precursors. Yeah, what is it actually on Earth? Probably not exactly the same as we've described it, but very, very similar. So this is a Miller and Urey experiment. I know you can't see that, but if you wanted to look into it, you could. So they did an experiment to simulate the conditions of early Earth with sea, a primeval kind of sea, an atmosphere of lots of methane and ammonia and water vapor, and they added some energy, some sparks. And as it circulated, they sampled it and found like lots of things that are organic, amino acids, hydrocarbons. So they found from inorganic precursors, amino acids, and hydrocarbons. So instead of forming in the atmosphere, however, the first organic molecules probably synthesized somewhere near submerged volcanoes and deep sea vents. So this is a deep sea vent along the mid-Atlantic ridge. It's very warm there. It's very hot, and the environment is conducive to the forming of oily droplets from organic molecules, and oily droplets, because oils attract each other, are able to form in water fairly easily. And that's what's thought to have happened. So small polymers, like little organic molecules, they polymerize when they're concentrated in hot sand or in a hot clay or rock. So in other words, they bind to each other, they come together. And protobionts are aggregates of abiotically produced molecules surrounded by a membrane or a membrane-like structure. So these are thought to be the small structures that predated cells. So protobionts, it seems, could have formed spontaneously. Small membrane-bound droplets called liposomes form automatically when lipids or other hydrophobic molecules are added to water. This is what they might look like. These are some actual liposomes. So this is an oily membrane, and they even form inside. Smaller droplets forming inside larger ones. So there may have been a very simple metabolism at first. If, for example, there are some enzymes included in the droplets that self-assemble, then some of them may carry out some simple metabolic functions. Yeah, so this is glucose phosphate phosphorylase. That's an enzyme which could form spontaneously, perhaps, and produce starch and maltose. And these are energy molecules. So this is, of course, this is hypothetical, but these conditions can more or less be recreated in a lab. Protobionts predating actual cells. So there's this idea that this was a question that came up quite often when the first origins of cells were discussed. So we talked about an enzyme. Well, you and I know that enzymes are proteins, and proteins are made from DNA code. But how can you get a DNA code if you have no proteins to copy the DNA? So that was a big question. Which came first, DNA or proteins? You can't really have one without the other, but they can't have both arrived at the same time. But it turns out that the first genetic material was probably not DNA, but RNA, probably RNA. And the reason that it's thought that it was RNA was because RNA molecules that we have now that function in things like cellular respiration, they're called ribozymes. And they catalyze a bunch of different reactions. So cell splicing, for example, making copies of their own sequence. So this is a molecule that you find in the body, a ribozyme, and it's instrumental in the process of protein translation or making proteins. So you don't really need a protein enzyme to do work or to synthesize some reactions. So perhaps the early protobionts, they were self-replicating. Catalytic RNA would have been more effective at using resources and would have increased in number through natural selection. And the reason why we think that is because if you look now at bacteria or viruses, particularly viruses that have RNA, they are more likely to mutate. So there is more variation likely in a world of RNA than there would be in a world of DNA. So that's a bit of an introduction to how the first cells might have formed. How the first cells may have formed. So the conditions were right. atmosphere, atmospheric conditions, plenty of water, inorganic molecules, heat, energy, yeah, that was part one. Part one, how the first cells may have formed from prehistoric Earth. Now we're going on to part two. Part two is about fossils. So index fossils are similar fossils found in the same strata in different locations. So there's, that's what Attenborough is going to be looking at all over the world, different strata. So because the continents were all together at one point and strata formed in a similar fashion, you will find similar fossils throughout the entire world. So that allows strata in one location to be correlated with strata at another location. So if you look at different locations, then you have more information and you can look at additional strata and start to see the relative appearance of various organisms on Earth. And how do you do that? Well, you can get the absolute ages of fossils as well. Let's see, I should write this down. Gives us the relative age of fossils. Or in other words, when one fossil might have appeared before another. So if we've got strata here, this might be a cliff. This is the lower strata. This is the upper strata. Well, these ones will be older. But it's just a relative kind of thing. However, you can get the absolute age by radiometric dating from the half-life of certain molecules. So isotopes decay into something else. Carbon-14, for example, decays into nitrogen, which is already 14. It's the main isotope of nitrogen. But carbon-14 is a radioisotope. So it will decay into nitrogen-14 because a radioisotope is unstable. So this graph here shows the number of parent isotopes. Say it's 1,000 carbon-14, and it'll start to decay into nitrogen. So in one half-life, you've got only 500 remaining parent isotopes. In another half-life, you've only got a quarter, so 250 remaining. But what you've accumulated is a daughter isotope, in this case, a nitrogen. So it will follow, will look something like that. So how else can we date different fossils? Well, one way is magnetic reversals. So every, I think it's something like 750,000 years or so, the magnetic poles reverse. So it's really neat is that the mid-Atlantic ridge, I'm pretty sure we've talked about the mid-Atlantic ridge before. Have we? I think so. That's a ridge down the center of the Atlantic Ocean, which is volcanic and is separating and making the Atlantic larger. So it's volcanic. And as such, it's adding to the crust. On either side. And if there are magnetic rock, then it will align itself to the poles. So it will align itself to the poles, say this is new material, and it aligns itself in this direction. But 750,000 years ago, everything was aligned in this direction because the poles were opposite. And so there's a change in magnetic orientation of magnetic particles like iron, for example. So that's really interesting. This gives us some dates, 750,000 years or so. So that's another way that you can date fossils. And it's been done for a long time, very, very early on. I think a chap that discovered the age of the Earth was not a scientist, actually, but it was his goal in life to discover the age of the Earth. And so he had to use a college's mass spectrometer in England and find the oldest rocks in the world, and did so. So you can study rocks and fossils at lots of different sites, and it's been done by many different scientists in many walks of life, and have come up with a geologic record. 3 eons, the Archean, the Proterozoic, the Phanerozoic, lots of eras and periods. I don't expect you to memorize them all, but some of them mark some major changes in the composition of fossil species. So here is a geologic record. And the only way I can show that to you close up is that's as close as I can get. The most interesting thing is when fossils appeared. So way back here, origin of the Earth. I can't write on it at the same time when it's blown up, but anyway, it's 4,600 million years ago. So 4.6 billion years. And the oldest known rocks, 3.8, 3.5 are the oldest prokaryotes. And then it took a very long time before eukaryote cells appeared. That was 2,500 or 2.5 billion years. And then it was a long time after that before any multicellularity appeared. So probably 600 or 700 million years ago. So the fossil record can chronicle a lot of large changes. Sometimes they were so rapid and so disruptive that a majority of species were simply swept away. So this is an example of various extinctions. 2 major ones, the Permian and the Cretaceous. So they're the ones that have received the most attention. The Permian, maybe 96% of marine animal species, and about 8 out of 27 orders of insects. They have disappeared then. It's thought to have been caused by enormous volcanic eruptions. And that predates the Cretaceous. The Cretaceous extinction, that was about 65 million years ago. That also caused a lot of marine and terrestrial organisms. And of course, we know most about the dinosaurs because they're talked about the most. They certainly weren't the only ones. Probably caused in part anyway by a large meteor. The climate was already changing. So there were already stresses on animals at that period and all organisms, plants and everything. But meteor struck the Yucatan Peninsula in Mexico, and it was enormous. And it caused a long-term climate change. It blocked out the sun, And so a lot of things went extinct then. Yeah, so we still need to learn a lot about mass extinctions. But what's interesting is that when there is an extinction and a lot of organisms disappear, it leaves room, it leaves vacated ecological niches and new ecological opportunities. So if the dinosaurs and other reptiles hadn't gone extinct, then mammals would probably not have evolved into the diversity that you see today. New ecological opportunities. So if you look at the geologic record as an analogy, as a clock, here humans are here. We appeared just before midnight, essentially. Yeah, so it's always fun to look at. Prokaryotes arrived here at around, so this is the origin of Earth. Prokaryotes about halfway through the life of Earth up to now. Yeah, 3.5 billion years ago or so. Eukaryotes here.
So it took about a billion years.
Oh, you scared me.
Sorry.
That's okay.
That was a little bit abrupt. I apologize.
No worries.
Yeah. I'm thinking of the Truman Show when there's the guy who the filmmaker, and when he talks, you can hear it from everywhere within the movie. Anyway, so multicellular eukaryotes started developing. Well, it took them about 1 billion years.
Yeah.
1 billion. Okay, before animals started to develop.
Yeah.
Okay.
Yeah. Exactly. It's quite a good. So here are animals here. Animalia. Land plants. Afterwards, because of course animals evolved in water. Yeah, that's a very good point. Whoops, where to go? Yeah, it's neat to look at because you can really see how old the Earth is and how young humans are. Very young. So as prokaryotes evolved, they probably exploited and changed the Earth quite a bit. You can tell there were eukaryotes because, or prokaryotes rather, sorry, because of structures that basically are remains of what they excreted. Yeah, so bacteria and sediment, calcium carbonate. I think these old fossils date 3.5 billion years ago. So this is Baja California. So it shows these mats that are basically bacterial mats. And this is Australia, Shark Bay. I got to see these, and boy, when you're looking at them, you're just imagining the amount of time that has passed since these organisms lived there. It just kind of blows your mind. But yeah, it's very interesting. So prokaryotes were around from 3.5 to 2 billion years ago. That's it. There's lots of different ways of making energy, electron transport. They were essential to early life, of course. Some possibly precede life itself. So the earliest types of photosynthesis didn't produce oxygen. But oxygenetic photosynthesis can be documented very closely because you can see it. Probably evolved 3.5 billion years ago. This is iron that's turned into rust because of oxygen. That's what oxygen does. Oxygen is very reactive and it rusts things. So that's a good sign of oxygen in the atmosphere. So yeah, when oxygen began to accumulate, there was more ecological opportunities. But it posed a challenge for life in that oxygen is very reactive. So if you are, as most bacteria probably were, anaerobes, oxygen would probably kill you. So it was quite a challenge. It did provide opportunity to get energy from light, and it provided organisms an opportunity to exploit new ecosystems, oxygenated ecosystems with oxygen. So eukaryote cells arose probably from symbioses and genetic exchange between prokaryotes. So prokaryotes definitely preceded eukaryotes by a long way. But that's a big question. How did eukaryote cells evolve from prokaryote cells? The oldest eukaryotes about 2.1 billion years ago. And somebody proposed a theory called the theory of endosymbiosis. The endosymbiont origin due to mitochondria and plastids. So they were probably small prokaryotes that were engulfed by larger prokaryotes. So it looks something like this. The prokaryote ancestors of mitochondria and plastids, they must have been ingested in some way. I know you can't see this very well, but here is a cell engulfing another cell. That's very good at supplying energy through cellular respiration. So these were the original mitochondria. And those original mitochondria had their own DNA, as all prokaryotes did. And as it turns out, if you look at our mitochondria, or any living organism mitochondria, it has its own DNA. So that is pretty good evidence that this could have been the way that eukaryotes first came about. And it turns out also that little photosynthetic prokaryotes were engulfed by others. And these are what we know now as chloroplasts that plants have for photosynthesis, and they also have their own DNA. And I'm mentioning it because it is a fairly strong evidence for that theory. So probably, possibly, the host which engulfed these organisms, the endosymbionts, would have become a single organism because they operated better together. And even now, there are some colonial organisms like Volvox. I don't know if you remember, we looked at that way back. That protist is colonial, but now people are saying, oh no, it's multicellular. So it just depends how specialized cells become in a colony. Yeah, so this is evidence supporting an endosymbiotic origin of mitochondria and plastids. That's like the chloroplast. They have very similar membranes, and they both have their own circular DNA. So there might have been additional endosymbiotic events, and also what's known as horizontal gene transfer. That's what prokaryotes do now, and that is how genes may be transferred. So, for example, you have a bacteria and a bacteria. I know we talked about this a little bit before. These are bacteria and horizontal gene transfer. So small plastids that can be transferred from one bacteria to another. And some investigators speculate that eukaryote flagella and cilia may have evolved from symbiotic bacteria. As bacteria now, nowadays, lots of bacteria have flagellae, and some eukaryotes also have flagellae. What about multicellularity? Well, it evolved several times in eukaryotes. So we have multicellular fungi, multicellular animals, animalia, multicellular plantae, but they probably evolved separately from unicellular to colonial to multicellular organisms. Yeah. So the very earliest ones date the common ancestor of all multicellular eukaryotes about 1.5 billion years ago. But the oldest known fossil, a relatively small algae that lived about 1.2 billion years ago. But larger organisms don't appear for a long time. So why could that be? Well, the conditions might not have been around much for fossilization, or they just haven't been discovered yet. But there are some 570 million year old fossils that were probably animal embryos found in China by Chinese paleontologists. And there is a connection definitely between a multicellular organism that were colonies. So I think I've talked about this before, and it makes sense to me that unicellular organisms, they do better if they combine in a colony because they all have different strengths. So one might be photosynthetic, for example, and one might be good at engulfing food. And one might be particularly good at reproduction. So these are all specialized cells. Eventually, they may only reproduce together because they're so dependent on each other. So then from there, so these are unicellular, forming colonies. They become inseparable and more specialized, and may then be considered a multicellular organism. Multicellularity. Yeah, so some cells became specialized in different functions, and some of those specializations, they had already appeared in the prokaryote world. But when we look at the film, I think Attenborough takes us up to the Cambrian explosion. So he's looking at a lot of things that I haven't mentioned in this talk. But this is when the major phyla of animals seem to have suddenly appeared. The first 20 million years of the Cambrian period. And so here you've got number of orders way down here. And all of a sudden you've got 60 orders of organisms. And the species within those organisms. So it is a period when at least we have fossils that have all the body plans of today's animals, today's animalia. Some are older than that, Cnidaria and Porifera. They may have dated to the late Proterozoic. And we looked at Cnidaria and Porifera. And you might have noticed the Porifera, for example, I think of them as kind of a glorified colony. So it makes sense, they're an older organism. And the Cnidaria are simple, I think they're like non-tissue organisms. So not organized into tissue yet. Quite simple. So we can look at molecular evidence, which is DNA, sequencing DNA. Lots of phyla originated and began to diverge even earlier, maybe 1 billion to 700 million years ago. Plants, fungi, animals colonized land about 500 million years ago. And there's lots of symbiotic relationships that have formed since then. They're common today, and they date from this time because living on land is no easy task. So if you have one organism that can provide you with shelter, like algae and fungus, for example, then you have a better chance of surviving on land. And we know that the continents are not fixed. They drift across the planet's surface on great plates of crust that float on the hot underlying mantle. So this is the faults here. I was just watching this morning. It's so interesting. Just looking at the types of civilizations. So this latest thing I'm watching, it's Ian Stewart, and what's it called? I think it's called History of Life, or History of Culture, or History of something like that. I'm not even sure exactly what it's called, but he's very interested in the change of geology and human civilizations.
I found this on the web by the change of geology. Check it out.
Oh, and civilizations tended to crop up along the faults between two plates. And it turns out that that's where water is very accessible. Yeah, so lots of geological processes occur at the boundaries, like subduction, for example. So here's one oceanic crust. It's subducting. And where it subducts, it's so hot down there that the rock melts and expands and pushes up into volcanoes. So the reason that you have a lot of coastal volcanoes is for that reason, or coastal mountain ranges is for that reason. And also they tend to buckle when they collide, and that causes other mountain ranges like the Rockies, for example, as a result of buckling. So here is, this is, I think, 250 million years ago. It's not really very long ago when you think of the evolution of organisms on Earth. So we were all just one big continent, Pangaea. It started to split up into Laurasia and Gondwana in the south. And it split up more 65 million years ago. Most of the continents were separate already until the configuration we have today. So there's lots of new information that revises our understanding of the tree of life, in particular molecular data, or DNA, and DNA sequencing. So we can sequence mostly just extant species, but we even have DNA from extinct species, like some dinosaurs and Neanderthal. But because it degrades, we often don't have an entire genome. As a matter of fact, I don't think anybody has an entire genome of an extinct species. So yeah, the early systems just had basically plants and animals. There used to be 5 kingdoms: Monera, Protista, Plantae, Fungi, and Animalia. That's the one I grew up with. But with the advent of molecular genetics and that, there's we organize our organisms in different domains. So this is a 3-domain system that's completely replaced the 5-kingdom system. And each domain is split into kingdoms. So this is one current view of biological diversity. This is what we looked at in chapter 27. We looked at prokaryotes in chapter 28, eukaryotes or protists. And so these are all the things that we've been looking at. The plants I hope to look at maybe by the end of the term, maybe fungi as well, I don't know. But we did cover Animalia pretty well, I think, in this term. So that's good. That's where I'd like to stop. Thank you for watching.